Uh, Good afternoon, everyone. So, welcome back again in my channel, Anatomy Circle. So, today we are learning the second part of the cerebellum, especially for the examination point of view. So, let's start with today's topic. So, yesterday we have discussed about the gross anatomy and classification of the cerebral hemisphere. Yes. So, in the cerebellum, we have also discussed about the its classification into three parts. So today we'll be discussing about the cerebral architecture, its connections, and the clinical anatomy associated with the cerebral hemisphere. Now, if you see the internal structure of the cerebellum, we know that the internal structure of the cerebellum, they are generally made up of the inner white matter and outer gray matter. Yeah. So today we have also discussed these things. And here, if you see in the middle, in the especially in the white matter part or inner medullary core part of the white matter, so you can see the four nucleus that are present in the cerebellar hemisphere. So they are the fastigial nucleus, globose, dentate, and emboliform nucleus. Yes, so these are the four nucleus that are present in the cerebral hemisphere or cerebellar hemisphere. Okay, that is the cerebellum. So Fastigi, globose, and Fastigi, globose, and emboliform nucleus. They are considered to be as a nucleus interpositor and the largest nucleus that is present in the human being. And in the primate, that one is a dentate nucleus. Okay, so if you see here, this one is a dentate nucleus. Now, in the internal structure of the cerebellum, so we know that they are made up of the inner gray, outer gray matter and inner white matter part. Yes. And here in the gray matter, what you will see is that there are the three layers that are present in the gray matter. Yes. So whatever you see this structure, this whole structure is a white matter and this structure is a gray matter. So especially here in the gray matter, what you will find the three layers. The first layer that is present here is called as molecular layer here, and the small layer, if you see here, this one is a Purkinje layer, and if you see this layer is a granular layer. Okay, so these are the three layers that are present in the cerebellum. And here in the inside, if you see this, all the structure, what you will find, this whole structure is a gray matter. Okay, so molecular layer, Purkinje layer, and granular layer. Now, after that, if you see the cerebellar histology of the cerebellum, so here what we have to understand is that there are many cells that are present here in the cerebellum. So basically, there are the five type of cells that are present in the cerebellum. Don't remember right now where they are present. Okay, just remember there are the five type of cells that are present in the cerebellum. The first cell is the Purkinje cell. The another layer is the basket cell. Another layer is a granule cell, Golgi cell, and Stellate cell. So these are the five type of cells that are present in the different layer of the cerebral hemisphere, especially in the outer gray matter part. Okay, in the molecular layer or in the Purkinje layer or in the granule layer, these five type of cells are present here. Okay, so we'll discuss where these cells are present later. Okay, now after that, if you see here in the molecular layer, okay, this one, the outermost layer is called as molecular layer. Okay, here in this molecular layer. So in the molecular layer, you will see which type of cell. So you will see the special type of cell, the two type of cell. That one is a stellate cell and the basket cell. So the stellate cell and the basket cell, stellate cell and the basket cell, these are the two main cells that are present in the molecular layer. Okay. And if you see in the Purkinje layer, yes, this middle layer that is present between the molecular layer and granular layer, this layer is called as Purkinje layer. So here in this Purkinje layer, you will find the one basket type or flat separate cell. Okay, and this flat separate cell is called as flat separate Purkinje cell. Okay, so Purkinje cell, and this cell they are always present in a row. Okay, in a single row. Ota row ma matra single. They are present singly. Okay, and after that, if you see in the lower part here in the granular layer, you will find the two cells. Okay, so this the name of the cells they start from the G. Yes, that is a granule cells or and Golgi cells. Okay, so sometimes these granule cells are also called as granular cells. Okay, so whether you see a granule cells or granular cells in the granular layer, they are same thing. Okay, and here and there are other different type of fibers are also present here. So we will discuss these fibers as we need it as our uh, slide progresses, okay, or as our class progresses. And one of the important things that we have to always remember here is that the term called as glomerulus. Okay, many times we use the term called as glomerulus. So especially here, there are a lot of fibers that are present. Okay, and there are two special fibers are present. The name of these fibers are called as climbing fiber and mosey fiber. Okay, so some of the part or the terminal enlarged part of the mosey fiber dendrites that are coming from the granule cells, yes, granule cells that are present in the granular layer and the Golgi cells. Okay, so these are forming a rosette separative structure or rose type of structure, and that structure is called as glomerulus. Okay, so please remember the term called as glomerulus. It is quite necessary the when you discuss the other slides. Okay, so remember this slide. Remember, we can remember this glomerulus slide. Okay, so this one is a cellular organism of the cerebellar cortex, showing the efferent and efferent fibers. So here, if you see in the cellular organization of the cerebral cortex, the the part that you see this part. Okay, so this part is considered to be as a three layers. Okay, molecular, Purkinje, and granular. Granular. Okay, so this one is a outer cortex part, or they are made up of the they are made up of the gray matter. Okay, so this one is a white matter. Okay, so gray matter it consists of three layers: molecular, Purkinje, and granular layer, and where you can see the different types of cells. So the different type of cells that are present, there are the five types, especially five type of cells and two type of fiber. Two type of fiber. Okay. Now, if you see, if you see in the flow chart as well, in the histology part, so we have discussed the molecular layer, Purkinje layer, and granular layer. So, if you want to draw the histology of the cerebellum, so you can just write this flow chart. Okay. So it will be quite easy to understand the flowchart. And whenever you discuss about the white matter, so this especially the white matter basically consists the neuroglial cells. Okay, so these are the supporting cells that are present here in the white matter. Here in the white matter. So in the molecular layer, there is a basket and stellate cell. Here in the Purkinje layer, you can see the Purkinje cell and the two G cells. Yes, granule cells or granular cells and Golgi cells, they are present here in the granular layer. So these are the five type of cells that are present in the cerebral slowly of the cerebellum or in the histoarchitecture or cytoarchitecture of the cerebellum. Now in the molecular layer, okay, so the molecular layer means one is the outermost layer that is present in the cerebellum. So in this layer, yes. So if you see here in this layer, in the molecular layer, so there are basically the nerve fiber that are present here in the molecular layer. They are unmyelinated nerve fibers are present here. And these nerve fibers, they are derived from the parallel fibers of the axons of the granule, stellate, and basket cells. So if you see this layer, yes, so this one is the stellate cell, yes, and if you see this one is the Purkinje cell. So basically, the Purkinje cells they are present mainly here in the Purkinje layer. But if you see in the molecular layer, you can see even the basket cells that are present here in the in the which layer? In the molecular layer. But Purkinje cell layer, that they are present in the Purkinje cell layer. And if we see in the uh, here, if you see, so some of the cells, you can see the granular cells. Granular cells, they also have the fibers that are coming from the, yes, this one is a granule cell layer to or into the molecular cell layer. Okay, so parallel fibers or the molecular cell layer, they basically contains the fibers that are actually coming from the stellate cell. Yes, these are the stellate cell fiber. These are the vascular cell fiber. And if you see this fiber that are actually coming from the granular cell. And in the Purkinje cell layer, if you see here in this cell layer, it basically contains the cell body of the Purkinje neuron. So these are somewhat looks like a flat sepet or basket like a cell, yes, basket type of cell. So basically we consider to be as a, or we can call it the shape of the Purkinje cell as a basket type of cell or flat sepet cell, okay? And if you see, they are arranged in a single row. They are arranged in a single row or in a single stratum, okay? And the dendrites that are present here in the dendrite of these Purkinje cell layer. So these layers, they are all the dendrites, they are present in the molecular layer, and they're making a synaptic connection here with the deep cerebellar nuclei. Okay, so dendrites, they are present here in the molecular layer of the basket cell layer, and the cell bodies of the uh, Purkinje cell layer that is present in the Purkinje layer. And if you see the axon, so axon, they are now connecting with the cerebellar nuclei, okay, or a deep cerebellar nuclei. Now, granular layer. So the granular layer is the third layer that is present between the Purkinje layer and the white matter. So this layer. Yes, so this there is a granular layer. So it basically contains the cell bodies and the dendrite of the two cells. That one is the 2G, yes, granule, Golgi and granular cell. Golgi and granular cell. So these are the two cells that are present in the granular layer. Okay, and the cell bodies you can see here in the third layer. So that is a granular layer. And their processes might be entering into the molecular layer. You may find in the Purkinje layer as well as. And if you see the granular layer and their processes, even they can be found here in the molecular layer. And sensory and motor fiber makes the synaptic connection. Okay, so we know that there are the two fibers that are the climbing fibers and motor fibers. So if you see the motor fibers now they are connecting with the processes of the Golgi cell as well as with the granule cell, and they are forming the glomerulus here in this region. Okay, so glomerulus is nothing, it's the motor fiber, Golgi cell and granular cell connection. Now, if you see the cerebellar cytoarchitecture, okay, in the cerebellar cytoarchitecture, we can study the cerebral cytoarchitecture on the basis of four things. Okay, the first one is an intrinsic neuron. Okay, the first one is a intrinsic neuron, and if you get confused with this photograph, we can even use a simple photograph or simple circuit. Okay, the same. These are the two equivalent photographs. Okay, in a very simple way. So it basically contains the intrinsic neuron, sensory input, output neuron, and intrinsic cerebral circuit. Okay, so basically we know that there are few neurons might be present there. There might be some might be the sensory input might be present there. There might be the output neurons might be present there, and intrinsic cerebral circuit are also present here in the cerebral cytotic. So we'll discuss these things. Okay. Now, if you see in the intrinsic neuron, okay. So intrinsic neuron means these are the cells. So there are almost almost 30 to 50 million of granule cells are present here in the cerebellum. Okay, 30 million of Purkinje cells. Okay, and 100 million of stellate and basket cells they are present here in the cerebellum. Okay, so these are the intrinsic neurons. These are the intrinsic neurons, and they are present in the different layer of the cerebral cortex. Now, in the Purkinje cell, if you see this cell layer, okay, so this basically contains the cell bodies, yes, cell bodies, Purkinje cell, and it looks somewhat like a flat sepet, okay, flat sepet, and lies in the Purkinje cell layer, okay, and that Purkinje cell layer is generated by PL here in the in the text, okay. So Purkinje cell that is present in the Purkinje cell layer, and if you see the dendrites of this Purkinje cell, so they form elaborating branches and they are present here in the molecular layer and extending into the granule layer as well. Okay. So they are extending into the granule layer as well. Now, and this granule layer, after extension into the granule layer, now the dendrites are terminating here into the deep cerebral peduncle or deep cerebral peduncle nuclei. Okay. Cerebral nuclei. And input for the Purkinje cell, now they comes from the granule cells, okay, granule cells and stellate cells, basket cells. You can see from the climbing fibers, okay, and as well as from the recurrent collateral, okay. So these are the inputs that are coming from the whatever the information or that information they are coming from the granule. You can see it's from the granule cell layer, okay, granule cell layer, and it might be coming from the stellate cell layer, and recurrent collateral might be there, as well as the climbing fiber might be there. Okay, so these are the information or efferent information that are coming to the Purkinje cell layer, and the output are they might be the lateral vestibular nucleus or recurrent collateral, and the output now they are entering into the deep cerebellar peduncle. Okay, so output now they are moving into the deep cerebral peduncle. And what is the importance of this Purkinje cell layer? Okay, so this Purkinje cell layer is a sole output neuron from the cerebellar cortex, and also exert a inhibitory influence to the deep cerebral and lateral vestibular nucleus. So it has a inhibitory influence on the deep cerebral and lateral vestibular nuclei. Okay, so Purkinje cell it is considered to be as the output neuron from the cerebellar cortex. Now, the granule cell, it is mainly present here in the granular layer. Okay, it is mainly present here in the granular layer, and the cell body, it is quite small and rounded. That basically contains 4 to 5 dendrites. Okay, uh, dendrites and mainly present in the granule layer, and it is this granule layer or the cell small rounded dendrites. Now they are getting connected with the Mosey fiber and dendrites that are coming from the Golgi cell and forming a glomerulus. Okay, forming a glomerulus. So these glomerulus, either you say these are the synaptic glomeruli or glomerulus. It looks somewhat like a claw-like appearance. Okay, claw-like appearance. So such claw-like appearance is called as glomerulus, where so where so it looks somewhat like a rosette shape also. Okay, so such type of shape is called as rosette shape. So such so claw-like appearance, or you can call it as a rosette shape. Okay, so granule, granule cell, Golgi cell, and Mosaic fiber they forming the glomerulus. So exons of the granule cell. Okay, so you see the exons of the granule cell. It moves above, and it reaches towards the molecular layer. Okay, so this one is also granule cell. It moves, the exons of this, it moves above and it reaches towards the molecular layer. And if you see the shape or of these axons, it bifurcates in the molecular layer, and after the bifurcation, they move into the opposite direction. Okay, so after bifurcation, they move on to the two opposite direction, and they are parallel with each other. Okay, they are also the fibers which are moving in the opposite direction. They are parallel with each other, so they are moving in the opposite direction after bifurcation. Yes, and moving in the opposite direction, and they are also parallel with each other. So kinetic contact. So what are the so while moving, so they get also coming they come in contact with the Purkinje cell. Yes. So if you see here, so they are coming in contact with the Purkinje cell. Yes. After that, they are also coming in contact with the stellate cells, and they are also coming in contact with the Golgi cells also. Okay, so if you see here, there is also the connection of the Golgi cells with the granule cells after dividing in a T-separate structure. Okay, so they have a connection with the other cells as well. Now, so what are the inputs that are coming or if efferents that are coming to the granule cell? So the efferents that are coming to the granule cell is the first one is a Moji fiber. Yes, the Moji fiber. So they are forming a glomerular cell. Okay. And other inputs, they are the collaterals coming from the climbing fiber. Some of the collaterals that might be come from here in this region as well. So they are forming the uh, input for the granule layer, okay, for this granule layer. So other are the exons that are coming from the Golgi cells, okay. So from here also that is coming, and some of the recurrent exons that are coming from the Purkinje cell, okay. So some of the recurrent, if you see here, so some of the recurrents that are coming from here, so they are also forming the input for the granule cell layer, okay. So here, if you remember, the glomerulus is the main input for the granule layer. So if you see the output layer, so the parallel fibers that are coming from here, okay. So they are the output fiber for the granule cell layer, okay. Parallel fibers. So if you see here, those fibers that are moving above here, so these are all the output fiber of the granule cell layer, okay. So So what is the main function of this granule cell layer? So this one is a excitatory neuron. Okay, you have to just remember granule cell layer is a excitatory neuron, and it is also loaded by the or its excitation is loaded by the Golgi and recurrent axon collateral of the Purkinje cell. So the excitatory neuron and its excitation might be loaded by the Golgi cell. Okay, so Golgi cell, this one is a inhibitory neuron. Now, stellate cells. So stellate cells, these are generally present in the if you see here in the outer part. Yes. So in the outer part, so if you remember the molecular layer, the outermost layer, it also contains the outer part and inner part. Yes, inner part. So molecular layer, the stellate cell they are present in the outer zone especially. Okay, and their cell bodies and processes may lie at the right angle to the axis. If this one is the axis, so their cell bodies they are making an angle of 90 degrees somewhere. Okay, so at the right angle. Now dendrites. So dendrites they are forming a synapse with the parallel fibers. Okay, so there are some parallel fibers that are moving here. Yes, that are coming from the granule cell layers. So they are forming the parallel fibers. So some of the collateral fibers that are coming from the climbing fibers. Okay, so they are also they might reach somewhere here in this region. They are also forming the collateral with the climbing fibers and some of the recurrent exons of the collateral of Purkinje. So here they are also making a connection with the Purkinje cell layer, and it looks somewhat like a arborized or branched out like a tree-like pattern. Okay, so such type of connections are made by the stellate cell. Okay, so generally present in the outer layer, making an angle of 90 degrees, so right angle to the axis and get connected with the Purkinje cell as well as with the parallel fibers that are coming from the granule, some of the fibers or collaterals that are coming from the climbing fibers, okay, and axons. Axons, they branch out like a pad, and their axons, if you see, it branches out like a tree-like branching. Okay. Now the basket cell. So basket cell, if you see here, so they are generally present here in the inner zone of the molecular layer. So in the molecular layer, in the outer layer, you will find the outer stellate layer. Yes, so stellate cell. But in the inner layer of the molecular layer, you will find the basket layer. So axons are right angle to the axis, and it covers or it characterizes the 10 to 12 Purkinje cell. Okay, so it's the covering capacity. Covering capacity means it can its axons get connected with the 10 to 12 number of Purkinje cells. Okay, 10 to 12 number of Purkinje cells. And it also making a kinetic connection with the pre-extant components of the Purkinje cell. Okay, so they are making a they are having a connection of 10 to 12 number of Purkinje cells. Okay, so if, so what is the importance of this basket cell? So both stellate cell and basket cell, they are the inhibitory neuron. Okay, basket cell and stellate cell both present in the molecular layer. It acts as an inhibitory neuron. Okay, and its inhibition. Okay, so whenever the excite whenever this stellate cell and basket cells, they get excited by the parallel fibers. Okay, whenever this get excited by the parallel fiber, okay, they suppresses the inhibitory response of the this Purkinje cells. Okay, so the excitation might be decreased by the help of Purkinje cells. Now Golgi cell. Now Golgi cell, now they are generally present here in the granular layer. Okay, so Golgi cell and granular or granule cell, they are present in the granular layer, and this is the largest neuron of the cerebral cortex. Okay, so this one is the largest type of neuron that is present here in the cerebellar cortex. Okay, and the cell bodies, they are present just situated outer zone of the granular layer. Okay, so again, if you divide the granular layer, this will be the outer zone, yes, and this will be the inner zone. So they are generally present in the outer zone. Okay, this largest neuron of the cerebral cortex, which are called as Golgi cells, they are present in the outer zone of the granular layer. Now, if you see the dendrites, so where the dendrites are moving here, so they are moving in the each and every direction. It is moving somewhere above as well as in the Purkinje layer as well as in the molecular layer. Okay, in the molecular layer they are moving above and they are synapsing somewhere here in this region. So these are nothing. So this one is the granule layer dividing into the T-shaped structure and forming the parallel fibers, yes, and they are making a connection with the parallel fibers. Okay, so there might be the climbing fibers might be coming here, yes, so they get connected with the climbing fiber as well. Okay, and some of the collateral fibers of the Purkinje cells might be present here, so they are also making a connection with the collateral fibers of the Purkinje cell right from here, this region. Okay, so they are making a connection with the all the type of cells. In the granule layer, GL means in a granule layer, it also synapses with the Moji fibers. Yes, so how they are getting synapses with the Moji fibers? With the help of this glomerulus. Okay, with the help of this glomerulus. So they are connecting and they are forming the synapse. Okay, and here you can see they also get connected with the granule layer here as well as with the Moji layer. Now axons. So axons, they are subdivided in the granule layer and come in contact with the granule cell. Okay. So here the Golgi cell. So axons may get divided in the granule layer and they may come contact here with the granular layer or granular. In the granular layer, there is a granule cell. So they are getting contact here in this region. Okay. Now inputs. So what are the inputs? from where the inputs are now coming into the Golgi cell. Okay, so the from the parallel fiber, from the climbing fiber, yes, as well as from the Mosey fiber. So these are the inputs from where the Golgi cells they are receiving its efferent or the information. Okay, and it might also get information from the recurrent axons that are coming from the collaterals of the PC. Okay, so that is the Purkinje cells. So right from here also they are getting the inputs. Output to the dendrite of the granule cell. So somewhere here in this region, yes, in the rho GT part, they are putting their information. So somewhere here in this region. So to the dendrite of the granule cell, they are making the output neuron. Or the output part or the output is tending towards the granule cell. Important. So Golgi cell, this one is also the inhibitory neuron. So whenever it gets excited, it gets diminished. The Golgi cell, the activity of this Golgi cell, it is this one is also the inhibitory neuron. So whenever it gets excited, okay, so the activity of this Golgi cell, it is inhibited by the granule cell. Okay. So now let's move on to the another part called as sensory inputs. Okay. So there are the two type of sensory inputs or efferents that are present in the cerebellum. So one is coming from the climbing fibers. Yes. One is coming from the climbing fibers and another is coming from the Mosey fiber. One is coming from the climbing fiber and another is coming from the Mosey fiber. But if you see these two type of cells, both are the excitatory neurons. Okay, they are the excitatory type, and they reach the cerebellum. They reach the cerebellum through the peduncles. Okay, they reach the cerebellum through the peduncles. If you see here, so there are the three type of peduncles. One is a superior peduncle, one is a middle cerebral peduncle, and another one is the inferior cerebral peduncle. The superior cerebral peduncle, they are actually responsible for the connection with the midbrain here in the above part, middle with the pons, and inferior cerebral peduncle, they are actually responsible for the connection with the medulla oblongata. Okay, medulla oblongata. So these are the three things. Now, sensory inputs. Let's discuss about the climbing fiber. Okay, so discuss about the climbing fiber. Now, if you see these climbing fibers, they are derived from now the climbing fibers, they are derived from the cells of the olivary nucleus. So, if you see the olivary nucleus, okay, so they are coming from the olivary nucleus and they are providing the collateral branches to synapse with the cerebellar nuclei. Okay, so they are providing these branches to get connected with the deep cerebral nuclei, and they make a monosynaptic contact means only the single contact. They are only making the single contact. Okay, or they are if they connect with the other type of cell, they are making only the single contact. So one climbing fiber only connect with the one type of cell. Now, on reaching the molecular layer, okay, so on reaching the molecular layer, it may synapse to the other type of neuron in the cerebral cortex, okay. So here they might reach to the molecular layer and they may get connected with the granule layer or stellate layer, or you can see along also with the Golgi layer, okay, Golgi cell, okay. So different type of cells that are present here in the molecular layer or the other connections that are coming from the granular layer. So these climbing fiber, it gets connected with those type of layers or cells as well. Now, the Mosey fiber, it gets synapsed with the numerous granule cell and some of the Golgi cell. Okay, so if you see here, the Mosey fiber, they are now forming the glomerulus. So now they are getting contact with the granule cell as well as the Golgi cell. So and they are forming the one very important structure called as glomerulus, okay, and is divided into the 30 to 40 terminal swellings, okay. So they are dividing into the 30 to 40 terminal zoning and getting in contact with the other type of cell. Now, climbing fibers directly excites the single Purkinje cell layer. Okay, so this climbing fiber, it might get excite only the single type of monocynetic. So because of the monocynetic condition, they can only excite the single PC or single Purkinje cell there. But whereas the mostly excites to the granule cell, okay, so because they are getting in contact with the granule cell layer as well as the Golgi cell layer forming the glomerular. So because with the help of this granule cell layer, okay, granule cell layer, it is again divided as a T-shaped structure forming the parallel structure, and they get connected to the Purkinje cell layer, they get connected to the Stellate cell layer, they get connected to the basket cell layer, they get connected to the Golgi cell layer. So at a time, the motif fiber, yes, motif fiber, it can fire the several thousand of Purkinje cell layer. Okay, at once it can fire the thousands of Purkinje cell layer. Now, output neurons. So, as you know that there are the four deep cerebral nuclei that are present here in the cerebellum. So, they are the output neuron of the cerebellum. So, they are the fastigial nucleus, global first, emboliform, and dentate nucleus. Okay, so here you can see these nucleus are the output neuron of the cerebellum. Now let's discuss about the intrinsic circuit of the cerebellum. So how these intrinsic circuits are formed. Okay, so there are the climbing afferent or climbing fiber and moji fiber. Now it gets connected to the deep cerebral nuclei with the help of some collaterals, as well as if you see here. So this deep cerebral nuclei means these are present in the white matter. Okay, this one is a white matter. Here in the white matter you can find these things, and this one is a outer gray matter. Yes? In the gray matter, you can see molecular, Purkinje, and granular cell layer. Okay, so this one is a gray matter. So climbing fiber, now it may reaches to the cerebral cortex, and you can see they are firing the Purkinje cell layer. Yes, the Moji fibers, they are also providing the collateral to the deep nuclei. They are forming the one of the structures of glomerular, and they can fire the thousand of Purkinje cells with the help of the glomerulus. Okay, so this is the intrinsic cerebral circuit that is present in the cerebellum. So this is a very small part. Okay, so this is how the intrinsic cerebral circuit are present. Okay, in the cerebellum. Now the efferent cerebral pathway. So efferent cerebral pathway basically contains the corticopontine cerebro olivocerebellar and cerebro cerebellar. Okay, so right from the different part of the cortex of the cerebellum, the information might be moving to the pons, yes, and now reaching towards the cerebellum. It might be coming from the olive nucleus and it may reaches towards the cerebellum. Again, from the cerebrum, it might use the reticular formation pathway and it may reach towards the cerebrum. So there are the different type of okay a pathway that are present in the cerebellum, okay, and they are controlling the different things, okay. So you can easily remember the pathway from the efferent cerebral pathway from this chart as well, okay. Similarly, if you see the efferent part, another efferent pathway, there might be the anterior spinocerebral pathway, posterior spinocerebral pathway. So if you see here, the spinal cord, they are now connecting with the cerebellum as well, okay, by the two pathway called as anterior and posterior cerebral pathway. There might be the involvement of QNO part, vestibular part, and other efferent pathway. They are also present here in the cerebellum. Similarly, if you see in the efferent pathway, okay, so there might be the involvement for the efferent pathway. There might be the involvement of the nucleus that are present in the cerebellum. That is the globus, emboliform, ruberal, dentothalamic, fastigial vestibular, fastigial reticular. So there are different pathways that are actually providing the efferent pathways. So they are controlling the different part in by the help of this pathway. Okay. So blood supply of the cerebellum. So in the blood supply of the cerebellum, so they are basically supplied by the, if you see here in this region, they are supplied by the superior cerebral artery. Okay, this one. If you see this one is supplying the superior cerebral artery, and after that, you can see the anterior inferior cellular artery and the posterior inferior cellular artery. So these are supplying the cerebellum here in this region. Okay, so we have to remember the blood supply of the cerebellum as well. Now, if you see in this photograph here, so these are the cerebral cross. Okay, so if you see this one is a pons and this one is a medulla oblongata. So to understand, so there are these are the borders that are present between the midbrain and pons. Okay, and if you see this region, if you see this region, so this region is considered to be as a part or the connection between the cerebellum pons, cerebellum and pons and medulla as well. Okay, so this part is called as cerebropontine angle. Okay, so the meeting point of the medulla oblongata, pons, and cerebellum is considered to be as the cerebropontine angle. So if there is any injury here in this region, and if you see here in this region, you can see the trigeminal nerve, and in the lower part, if you see it, you can see the facial nerve as well as the vestibulocochlear nerve. So if there is a damage in this region, so loss of hearing, equilibrium, maintenance of the equilibrium or adjustment, and because of the paralysis of the facial muscle, the facial paralysis might also occur if there is injury in the cerebropontine angle. Similarly, there might be the tumor again here in this region. Okay, so that tumor is also called as cerebropontine angle tumor. So here in this region. Okay, so small triangular interval bounded by the pons anteromedially and cerebellum posteromedially. So that where in the cerebropontine angle, if there is a tumor, we consider to be as cerebropontine tumor. So again, there might be the involvement of the trigeminal nucleus. So involvement of the trigeminal nerve cause the involvement of the spinal nucleus of the trigeminal nerve. So involvement of this spinal nucleus leads to the loss of the sensation of the pain and temperature over the face. Okay, so because of the involvement of the face as facial nerve as well here in this region, so tumor here in this region might get compressed the trigeminal nerve. So if there is a compression of the trigeminal nerve, so there will be the compression of the spinal nucleus causing the loss of sensation of the pain and temperature over the face. Similarly, there might be the involvement of facial nucleus as well, so facial paralysis. Middle cerebral peduncle here causing ataxia. Fibers of the nucleus of the vestibulocochlear nerve causing vertigo, tinnitus, nystagmus, and deafness might be also present here. So, vermis. So, medulloblastoma of the vermis in the children is called as, so if there is a tumor here in this region, okay, or here in this region in the vermis part, so that is called as vermis syndrome, okay. So what happens in this Warnick syndrome? So tendency to the person starts to fall backward or forward, difficulty in holding head steady in the steady position, okay, and difficulty in holding the trunk erect. That is also the sign and symptoms of the Bormis syndrome. So because of the tumor that is present here in the medulla part, yes, in the medulloblastoma of the Bormis part, especially. So another tumor might be present in the cerebellum, in the one of the part of the cerebellum. Yes. So that might lead to the cerebral hemisphere syndrome. That might be present unilateral, especially in the one side only. So muscles of one side of the diseased cerebral hemisphere might get involved, uh, falling to the one side, yes, delaying in the initiation of the movement. So these are all the symptoms of the cerebral hemisphere syndrome. Okay. So today we have discussed about the cerebellum as an exam point of view. So we have discussed about the cytoarchitecture part, especially we have discussed all the cells that are present in the cerebellum. So especially the five type of cells. Few are excitatory and other are inhibitory type of cells. Yes, and we have also seen the efferent and efferent pathway along with the circuit. So thank you so much for listening and listening this class. And if you have any queries related to this topic, I need requesting to add your question in the comment box. Thank you so much.